let me kind of start with the recording. So lab practical one review. Basically what you will see, you will have a picture popping up on a lab practical, and that particular picture, it will be either some sort of questions regarding the frequencies, formulas, etc. And this is the sample questions, or it might be the question regarding any particular specimen from our lab. I'm trying to keep all of the lab practical images very similar or pretty much identical to whatever we covered in our lab. Again, that's the first lab practical and the second lab practical. Third and fourth has been created by the other professor, and the images are slightly different, but I'll see if I will be able to update those ones. All right, so basically you already have the review for the lab practical. Make sure you will be able to answer those particular questions. Before we'll go into the questions, let me show you a quick website. That's a Flickr. So you're basically going on the Flickr and you are typing my name. So let me pull out something like that. All right, so let's do this. Going flickr.com. Here it will load it up. I'm not logged in. For Flickr, you don't have to have a account. It's free. And what you will do, you will type my name, Igor Yakushkin, and then very important, you will search for people. If you look for the photos, I don't believe I'm there. Let's see right there, not. But if you'll choose people, it will give me my my link, and then you can click on it and you'll go to albums. I'm updating right now. At some point of time, Flickr was allowing unlimited amount of images. Now they have cap you at 1,000. Everything else is paid. So anyway, this is lab practical one for bio one, and that's a bio one again, and that's a bio one again. But starting from here, you can see bio 2, lab 2, hominid skull. If you click on it, you will have our hominid skulls right there, and so on and so forth. You can click on each one of those if you want to. Gives you a little bit of description. In some cases, well, gorilla, it's kind of gives you the skull itself, and so on and so forth. And periodically you will have the ads, and I'm not receiving money whatsoever, not a penny. For that, it's just for maintaining those 1,000 pictures, I guess. So we have all of those. This one is available to you, and basically it's very similar to what you already have in the lab guides. But if you want to go, please use it. Here we have bacteria, and it's talking about the stromatolites, and it gives you the idea of the bacterial cell. So pretty much everything what we covered before. And once again, periodically it tells you what it is. Bacterial cell, gram negative. Bacteria cell wall, gram positive. All right. So that's the endospore, right? Yeah, bacterial spore or endospore. Here we have the models. So, and there it says gram negative spirochete. So if you want to, feel free to use it. It's one of those particular additional things you might consider. Generally, I did it or made it for face-to-face classes when I didn't have the guides. But again, it looks very similar to what we have on the guides and so on. I will expand it a little bit. I will be working. First I will need to delete some of the extra pictures, and then starting with, like, say, plants. This is the plants. I will be adding few things to it. But again, I'm capped at 1,000 and I'm already at 933. So anyway, that's just kind of like one of the additional things which you might want to use it. Up to you. And again, it's friendly. You can go from a Mac, you can go from the cell phone, you can go from the tablet. Doesn't really matter. It's a website. They also have the app, which is all free, and you don't have to have the account. I have to have account in order to go with that. But it's again one of those particular things which might help you with the lab practical. Let's see, review. So again, in equation, P plus Q equal 1, know what is the frequency. So if the question is asking, for example, in a P plus Q equal 1, P represents blah. And you need to say not the dominant allele. It does not represent the dominant allele. It's a frequency of dominant allele. Same thing with a Q, but it's a frequency of recessive. Same thing with a P squared, QPQ, Q squared, in that particular equation, frequency, and so on and so forth. Now, remember the genotype. What is the phenotype? And we're talking about the bunny lab or just general. That's the dominant homozygous phenotype. That's a heterozygous phenotype. Most likely will show up as a dominant. And that's the homozygous recessive phenotype. And if we're talking about the bunnies, that would be furry, that one would be furry, that one is furless. How did the environment change in generation 4? That's referring to our lab. Remember the furry third ones. And then in the generation 4, remember we added the natural selection or directional. Now we have a specific direction. The nature is selecting the furry one, and the furless ones would be dying off. And again, Hardy-Weinberg equilibrium, same as the test. Make sure that you know those, several of them, not all 5. Well, technically you should know all 5, but if you can list, it might say list 2 or list 3 conditions for the Hardy-Weinberg equilibrium. Any questions, any problems so far? All right, so make sure that you understand that if the alleles are not changing, remember this one, alleles are not changing. Alleles not changing, it means that the population is in equilibrium. Equilibrium between the frequency of P and Q, same thing, right? 57 and 43, 57 and 43, 57 and 43. Meaning that there is no evolution. We talk about that, we tied it up to the evolution. So if there is no evolution, there is no changes in frequency. Or in the reverse, if there is a evolution happening, the frequency would change. That's one of the main idea why we're using that particular equation to calculate and then compare to what we have in real life, so to speak. So reference point. And again, which one would be opposite? So if they maintain the equilibrium, you should not have no mutations, no natural selection, huge population, random mating without any preferences, so non-preferential mating, no genetic drift, no immigration, emigration. Here you should say, all right, well, natural selection occurs, mutations occur, non-random now, selective mating. And then the population is small, and we have some sort of immigration into or out of the population. And then, of course, we have a genetic drift. All right, that's basically all what we had. And make sure that you will be able to answer the post-lab questions. And remember, we had those particular problems. If Q square is 0.49, can you find the P? Can you find the Q? Can you find the 2PQ or anything like that? And then the percentages. Remember, we did that part. So here, something like that. If you revisit the population, so here you're looking, calculating the frequencies, and then you're comparing to the next generation. So make sure you will be able to do all of the post-lab problems. Overview them. They are on the lab practical, similar. So if you have any questions, you're not really sure how to solve those problems, please let me know, okay? Shoot me a quick email saying, hey, I don't really remember how you ended up with such a number. All right, well, lab number 2 was about the primates, and we talk about the different species, great apes, as well as the australopiths. And we talk about the gorilla and chimpanzee, that's the modern species, and then the ancient species, the Australopithecus, and then actually the Achadensis. So all of those, make sure you will be able to identify five major structures: sagittal crest, brow ridge, zygomatic arch, snout, and then on the bottom we have the foramen magnum, right? So again, sagittal crest, zygomatic arch, brow ridge, snout, and the foramen magnum. So yeah, brain case, remember in the evolution of the hominids, including humans, one of the main branches which we are on, the main idea was increasing of the brain case, right? From somewhere around 450 to 500 cubic centimeters, it started raising the size up almost 3 times, and we are right now standing at about 1450, 1500 cubic centimeters of brain space. As a result, our jaws decreased, teeth decreased, zygomatic arch much smaller, no sagittal crest, flat face. Regarding those particular species, make sure you know the skulls, how the skull looks like, and you will be able to approximately place it into the timeline. I mean, this one is older, this one is the middle, this one is younger or youngest one, right? So that one, do I ask, well, let's say I would not ask you, name the Latin species of that skull or name the Latin species of that skull, but you need to be able to recognize 3 major genus on our branch. One of the genus is Australopithecus. There is 2 species. Then there is a Paranthropus with the 3 species, but you don't have to worry about the species. And then the rest of them, I don't really remember how many. We talk about Habilis, Ergaster, Erectus. Presumably it's 2 different Hilderbegenesis, Neanderthalensis, and the Homo sapiens. So we have 6 species of genus Homo, means human, right? So make sure that you will be able to know those. So here we have the Australopithecus, then another Australopithecus afarensis, did that. Then we have 3 species. There is more than 3 species, but we covered 3 species of Paranthropuses as dead end near the humans, very massive skulls, very large bones, jaws, very pronounced brow ridge. In most of the cases, you can see the sagittal crest. So they went all muscles. Our ancestor went all brain, so to speak. So in this particular case, yeah, Paranthropus. So make sure that you will be able to recognize the genes. Yeah, Paranthropus. And if you'll make a tiny little bitty mistake in the spelling, as long as I understand what you're talking about, I usually don't discount on the misspelling, because all of them will be short answers. Basically, you'll have to type it in something. So here we have, and finally Homo species. So yes, we had 6 species. There is 7 skulls, but the Cro-Magnons and modern humans, that's a Homo sapiens. So we have Habilis, we have Erectus, Ergaster, Hilderbegenesis, the Neanderthal, and 2 Homo sapiens. So yeah, Habilis, relatively small, kind of looks similar to the chimpanzee skull. It's not a chimpanzee, as you can see. It has a much more human characteristics, but smaller skull, flat face, bipedal, then the bigger skull. Again, we're still looking at the fairly strong brow ridges and fairly strong jaw. But then in the ergaster, what we see is the brow ridges are receding, still have them, and the jaw become much smaller with the smaller teeth. Why? Because they start cooking food. Well, change the diet from the predominantly vegetarian, went into a omnivorous diet. So they start including meat, actively hunting, and start cooking. And we'll talk about this guy, Phildarbigenesis. This one is most likely, if you'll see the guy sitting right next to you on the bus or any kind of public transportation, subway or anything like that, you're like, kind of weird looking one, but other than that, it was very, very human-like. And then, of course, yeah, the Neanderthals, you probably will, again, very similar to humans, very close related species to us. And then the Cro-Magnons, if you take that particular guy, kind of wash it, put some makeup on, you will not be able to tell the difference between the regular lady and, well, human sapiens versus, well, it is a human, a Homo sapiens already. So, yeah. Anyway, so next one are the bacteria. For the bacteria part, make sure you will be able to recognize all of the shapes. And we're talking about how many? 4 major shapes: coccus, bacillus, vibrio, and spirochete, or spirillum, either or. Then make sure to be able to recognize on the model cell membrane and cell wall. Remember what this cell wall is made out of: peptidoglycans, both of them positive and negative, but negative in addition to the peptidoglycans has the lipopolysaccharides. And then, of course, the flagella or flagella plural, flagellum singular, the chromosome. That's basically what you will need to be. Know that the stromatolites are the fossilized autotrophic algae, well, bacterial essentially. So here we go, how the stromatolites look like. And then the bacterial cell, we already talked about the cell membrane. Let me kind of close up. So here we have a cell, bacterial cell, the chromosome, the cell membrane, the inner part, cell wall is the blue part, and then the outer wall is the capsule. This stuff sticking out known as the pili, the short projections, the long one is the flagellum. This is the chromosome inside the cytoplasm. The pink dots are ribosomes, and those kind of blue circular thingies are plasmids, the pieces of DNA outside of the main DNA of the bacteria. So that one, yeah. Here we have the spore or endospore, and here we're looking at the 2 cell walls, right? This is the cell membrane, this is the cell membrane. Now we're looking at the cell wall. That one is pretty much completely made out of peptidoglycans, so it's a gram-positive cell wall. Here we have a thin layer of peptidoglycans and then lipopolysaccharides, so that would be a gram-negative cell wall. Remember, gram-positive supposed to stain, well, the technique, staining technique should be staining the wall bluish purple. If it's not bluish purple, usually pink, it will be gram-negative. But treponema is actually kind of brownish reddish color, so, but it's still not bluish purplish color. 2 autotrophic, so here we're looking at the gram-positive cell wall. Now we're looking at the gram-negative cell wall. The models, the cocci, the rods, well, the cocci would be round ones, the rod ones, right here, right there, all of those are rod style. That's the bacillus, and then we're looking at spirilla or spirochete right here, and those kind of sausage looking one, they are the vibrios. All right. So here we have some of the Vibrio again, comma-looking Spirochaetospirillum. Bacillus is the rod one, is just the chain, and the round ones, which are the cocci. And the spore destruction inside of the bacteria. Also, if you'll see looking like this right here, this is the endospores right there, those brown ones and not the white ones. So heterotrophic, we looked at this is the chains of anthrax, and anthrax are gram positive, and that's the, if you kind of increase the size, you can see that this is the rods. So that's a bacillus, right? Next one are the gonorrhea, and those ones are clusters of tiny little bitty round things. So that would be cocci. Here we have a treponema. As you can see, it's kind of brownish reddish, and that one is the spirochete. You can say it doesn't really look like a spirochete. Well, it is, because if you see, let me find a good one. This one, the good one. Let's see. Yeah. This one, you can actually see that it's actually a very, very thin spiral. All right. So that's the treponema. But again, on the lab practical, I'll try to put something which is very obvious. That one, for example, is very commonly seen on the lab practical because you can clearly see it's a spirochete, right? And all of them are heterotrophic bacteria. Again, all of those that are gram negative bacteria. Here we have the comma shape, the Vibrios. Again, under high magnification, you can see the comma shape of the bacteria, and also they are pinkish color, so gram negative. You can clearly see the comma right here, a bunch of them. So that one is cholera Vibrio. All right, autotrophic bacteria, we talk about the oscillatoria, right here. And in this case, I think I have a higher magnification. So you can see it's basically a cell right next to the cell, right next to the cell. If the cell dies somewhere in the middle, it will lose the chlorophyll, not chloroplast. It does not have chloroplast. Remember, it's all bacteria, so prokaryotes. But they have a chlorophyll as the pigment, so whenever they dies, a single cell died. So now we're looking at the darker spot known as the dead cell, also known as the separation spot. So, and you can see it's basically a colony of the bacteria. Here we have probably 2 bacteria died, and so on. And here we have anabina, kind of the round ones, so they're round, and they creating the chain of those as well. So we have the, for the oscillatory, be able to identify the filament, basically the strand. Vegetative cell is one of those, and then the dead cell is the black part. What type of substrate do you use to grow bacteria? Oh, that's the next one. Growing the bacteria, what kind of substrate? Agar, right? Or agarose, this particular semi-transparent gel. And here we have some of the bacterial colonies. It's not a single bacteria. Please make sure you understand that. That's not a single bacteria. It's like billions of bacteria, but it is a clone of the same bacteria, so we can start seeing those ones. And they, of course, will be slightly different because, well, we will have a different species, different colors, different shapes, and so on. So yeah, definitely can clearly see. Here we have one colony, which is round. This is another colony, different species, most likely because it's actually growing into the agarose, meaning they not really required much of the oxygen. Here we have a yellow one. The fuzzy ones are most likely mold. You can actually start seeing that the sporangia produced, sporangia being produced, we have a few other colonies of the bacteria. Initially, it was one bacteria, and then it started growing, so we can start seeing. What is the optimal bacteria growth temperature? Body temperature at around 36, 37 degrees Celsius, roughly 95, 97 degrees Fahrenheit. Do they produce sexually or asexually? Those particular bacteria, they were going through the binary fission, means they reproduce asexually. But again, bacteria has several ways how to reproduce sexually as well. And I think we are kind of done with the lab 3. Now lab 4, let me pull it out. Lab 4 are protists, and in the protist, what we have, again, we have 3 different means for locomotion: flagella, pseudopods, and cilia right here. So here we have amoeba produces the pseudopods, and within the protist, now it's a eukaryotic cell, unlike the bacteria. So it has the nucleus, right? It will have mitochondria, definitely cytoplasm, definitely cell membrane, and that one would be a contractile vacuole. And the pseudopod, of course, contractile vacuole expel the excess water. So now we're looking at the nucleus, cytoplasm. Yeah, talk about that. And this is how the amoebas are looking under the microscopes. I had students before asking me why they all different colors. Different staining techniques has been used. As a result, you can see them because under the normal circumstances, they are semi-transparent, basically kind of grayish. Yeah, here we go. That's the no staining amoeba. This is how it will look like. It actually was a live amoeba when I was taking the picture of them. So now Cilius, a paramecium, very, very fast moving, has 2 nuclei. The large one macronucleus and the small one micronucleus, and we have cilias all over the body, so they're really fast, and because they are moving into the environment, exchanging the nutrients with exchanging the substances with the environment, they require 2 contractile vacuoles. So we have 2 of those particular star-looking shapes. So this is how it closed, and now we're looking at the open one. This is the contractile vacuole channels, and the contractile vacuole is actually that it will increase and then expel excess water. And we're looking at the large macronuclei, and the small one is right here. Small ones used for sexual reproduction. For everything else, macronuclei taking the main role or using the main role, and you can see the cilias right here. So this is again stained parameciums, different magnifications, the smaller magnification, higher magnification, and this is how they look under a normal microscope without any staining technique. You can see actually in some of them you can see the cilias right here. So yeah, and that particular white spot is most likely it's a large contractile vacuole. And lastly, we have the euglena. Euglena is phototrophic. Before amoeba as well as the parameciums are heterotrophic. Euglena is autotrophic, or you can say mixotrophic, because if left without the light, it will go and start actively hunting for smaller creatures such as bacteria and smaller protists. So let's take a look. On that particular table, we have amoeba, paramecium, euglena, and trypanosoma, all unicellular. Motility means what? Here we have a pseudopod, paramecium, cilia, euglena, flagellum. Trypanosoma, you can put 2 things, either flagellum, and I will be okay with that, or you can put undulating membrane, because since the trypanosoma lives in the blood, it needs an extra surface in order to move better. So now amoeba heterotrophic, paramecium heterotrophic, euglena is autotrophic / mixotrophic. Either or will do. But if you'll answer heterotrophic, euglena is not just the heterotrophic. It's either mixo or autotrophic. And the trypanosoma, since it's a parasite, it's the heterotrophic. Free living, free living, free living, and the trypanosoma is parasitic. How trypanosoma spreads? Remember, tsetse fly bringing, well, basically biting the infected person or infected organism. Usually it's not the person, it's most of the time it's large hoofed animals in the African savanna. It bites, feeds on the infected blood, becoming infected, going further and bite another animal, and whenever it's injecting it with its saliva, the parasite goes into the blood. So do the protists reproduce sexually, asexually, or both? The answer is both, right? We already talked about the mitosis, and we talk about the sexual reproduction. Describe how paramecium reproduce asexually. So here we're looking at the euglena. Euglena actually under the microscope, as you can see, they are drastically smaller than the other one. On this one, you can kind of see, the flagellum, very thin, and it's constantly beating. So under the microscope, it's kind of hard to get it. But you also can see that red spot right here. So this is where the flagella located at. So that's the front end of the creature. So here we're looking at the slide of the trypanosomas. White blood, red blood cells, all of those pink circles, that the blue one would be the white blood cell. Again, why? Because of the staining and the squiggly parts, wormy looking structures. It's not worms, it's a protist, it's trypanosoma, a single cell protist infecting the blood of the, in this particular case, that's the mammal. And this is how it reproduces. See, tsetse fly is the vector. This is how a mammal can get it from another mammal. If, let's say, a human who has the trypanosoma will, let's say, sneeze or cough on you, nothing going to happen. Have to be blood transfer from the infected to non-infected person. So here, once again, trypanosomas, red blood cells. Paramecium. Again, if you'll see the picture with one cell splitting into two cells, that's asexual reproduction, also known as the mitosis first and then cytokinesis. You can put it as a fission. I prefer for you not to use the term, even though I think lab is talking about binary fission. Binary fission, we're usually talking about the bacterial division. Here, a fission will do. I might accept the binary fission, but try not to use it. Mitosis is a better choice for that. Okay, so again, we're looking at the one cell going, splitting into two cells. Yeah, asexual fission will do just fine, but try not to say the binary fission. Binary fission is most likely for the prokaryotes. So here we're looking at the actual slides. Now we're looking at the sexual reproduction. Why? Because we're looking at the two cells merging. So here we have two cells merging. This is the two cells, and that's the two cells. So here you clearly see that there is a merging of the cell membranes, and soon they will be exchanging the micronuclei. Again, we're looking at the fission, fusion, merging of the not bacteria, parameciums. So that's the eukaryotes, right? So in this particular case, a reproduction asexually. And the question might say, is it a sexual or asexual reproduction? Well, you'll see too, that's definitely sexual reproduction. All right, now we going, so yes, reproduction, we talk about that. Now, chlorophyta. Well, you already had that particular table filled up and during that particular lab, but let's kind of say common name of the group. Green algae. So this all is green algae, right? Now, phaetophyta is brown algae. Rhodophyta are red algae. Chrysophyta are golden algae. Spirogyra is multicellular. Volvox is colonial. Ulva is multicellular. Sargassum and Nereocystis are multicellular. Porphyra multicellular. Diatom single cell. And any kind of characteristics you need to write down for you, not for me, for yourself, so you will be able to recognize them whenever you'll see the picture of the Volvox, for example. Like, looks like a sphere. Keep in mind that this is something which known as adult colony, and the smaller ones known as the daughter colonies, right? There might be an arrow saying name the structure, and it's pointing at the daughter colony, right? So you can, this one, for example, this is all adult colony, and we have 1, 2, 3, 4, 5, 6, 7, 8 daughter colonies inside. As they grow, they literally will open up, break apart the adult colony, and you can see the part of the broken adult colony already, and they will be released. That's the asexual reproduction for the Volvox. And do they have the sexual reproduction? Yes, they do. We're not going there. So yeah, adult colony versus daughter colony. So here we have a Spirogyra, and Spirogyra in this particular case, 2 filaments laying right next to each other, and you need to be able to recognize which one is a male, which one is a female, based on the presence of the darker structure which known as a zygote. Remember, male and female, here we go. Male and female are male and female. It's not entirely correct statement, but males are generally donating, females accepting the DNA. So wherever you will see the darker spot, zygote, the combination of the DNA from male and the female, that's the most likely female filament. Therefore, that guy would be a male, and you can actually see the transfer of the genetic material in process. But kind of on a static picture, kind of hard to say which way it goes, but you can clearly see that here we have the zygote. So that's the darker part is the zygote. So yeah. So here we go. So in this case, left is the male, right is the female because of the zygote right here. So, and this is how the dried up Spirogyra look like threads, tiny little bitty threads. All right. Now, whenever you're looking at the vegetative strand, you can clearly see the strand, right? The nuclei, and that particular spiral is nothing else but the chloroplast. And then, of course, we talk a little bit about the pyrenoids. Whenever you're looking at the larger magnification along the chloroplast, you will see those dark round spots. That's the pyrenoids. This is basically accumulation of the starch. Where the starch coming from? Remember, photosynthesis, right? Carbon dioxide, 6 of them, plus 6 waters, plus sunlight will give you glucose, C6H12O6, plus 6 oxygens. So in this particular case, whenever you're taking glucose, combined with the glucose, creating the starch, well, chain, that turns into either starch or cellulose as well. Cellulose is made out of glucose as well. So here we're looking at the really nice pyrenoids, a lot of them, right on the circular or spiral chloroplast. So again, this is how this Spirogyra looks whenever it's dried up. Okay, here we have a sea lettuce, also known as the ulva. You can clearly see why it's called sea lettuce, so green algae. So now the macrocysts, right, here we go. So on that particular slide, you'll need to be able to identify the blades, and the function of the blade is photosynthesis. Now we have the hold fast, basically roots, they're not roots, they just holding the whole body of the algae at the at place, attaches to the substrate, bottom of the ocean, so to speak. And then we have this type. It's similar to the stem, but not stem yet. We'll talk next unit what considered to be a stem. And this particular structure known as the float. So it brings the blades closer to the surface. So in this particular case, more sun, more sunlight. More sunlight means faster the photosynthesis. More photosynthesis means more food. So make sure that you know, be able to recognize and know the function, blades, type, hold fast, etc. And we talk about the sargassum. They are lacking of the hold fast. That's why they're just floating on top, because they have tons of different tons of tiny little bitty floats. As a result, it allows the whole algae to float on top of the waves, essentially. Yeah, here we have the uses. If we are either eating it or using it for medicine, right? Rhodophyta, as well as the Chrysophyta. So here we're looking at the Porphyra, and Porphyra is kind of reddish, has a reddish tint to it, because once again it has the reddish, it's using a different set of the pigments. Green algae is using the chlorophyll, red algae, in addition to the chlorophyll, they're using a red pigments. And then we have a diatoms. Of course, we talk about the diatoms. Diatoms are made kind of out of two parts, or shell is made out of two parts, hence the name. Di means two. And then the shell is transparent, made out of silica, silicon dioxide, essentially glass. And they come in very intricate forms. I might have a radial symmetry, all kinds of neat stuff right there, but we're not going too deep into that. Any questions, any problems at this moment? All right. Well, you guys are not very active today. I guess too tired from the test, huh? All right, last lab. I almost finished though. Last lab was the lab number 5. Let me pull the guide for it really quickly. All right, here we have the fungi and lichens. So again, remember, all of the classification is based on the structures for sexual reproduction. We're looking at the 4 major phylum: Zygomycota, Basidiomycota, Ascomycota, and Deuteromycota. The sexual sporangium will be called zygospore, right? Zygosporangium, Basidia, Ascus, and the Deuteromycota don't have, well, we don't know any sexual reproduction for that particular group. So Zygomycota, also known as a rhizopus, you can, it's a black bread mold, but this is the model. And here what you will need to be able to identify are the strands known as a hyphae, the combination of the strands known as a mycelium, the sporangium, and you need to be able to recognize which one is which one is the sexual sporangium, which one is the asexual sporangium. In this particular case, the top is exactly the same. You need to take a look where it's growing out of. If it's growing out of, straight out of mycelium, essentially out of the hyphae, it's asexual reproduction. If it grows from the zygote, right here, that particular round brown structure, this is a zygosporangium, which will produce sexually reproduced spores. Spores are always what? Haploid, right? So keep that in mind. So here we have asexual and sexual sporangium. This is the zygote. So here we have asexual, sexual, and the name of that particular mold is rhizopus. So under the microscope, you need to be able to recognize, is it a sexual or asexual reproduction? Well, you clearly can see that one strand, another strand coming in, creating that zygote. As soon as you're talking about zygote, you're talking about exchange of the genetic material, so that's a sexual reproduction. Here you clearly see that one and another strand connecting, creating the baby. And that particular baby is the genetically different from this side and that side as well, from positive and negative strand, and that will soon sprung out the sexual zygote sporangium. Here, however, you're looking at this sporangium and it just kind of comes out from a tiny little bitty strand. So there is no exchange of genetic material, so it's an asexual reproduction. All of the spores produced here will be genetically identical to each other and to the strand it produces. So here we go. But as you can see, tons of them produced, billions of spores to cover as much of the area as possible. So it's kind of talking about the formation of the zygote. So basically we're looking at the sexual reproduction right here. So yeah. Now here we're looking at the basidiomycota, also known as a club fungi, because they have a structure within the cap, usually within a cap, where 4 spores will be produced. Spores will be produced by meiosis, therefore it's a sexual reproduction. Basidiocarp. Basidio, again, carp is the body which contains basidia, essentially a mushroom or shelf mushroom. That's the basidiocarp. You can also call it sporocarp, the body producing spores. Basidium and a basidia spore. So here we're looking at, this is the mushroom, right? But the scientific name for that would be a basidiocarp, because this is the structure which takes out of the ground and spreading the spores. All that particular white stuff are mycelium, which is embedded within the substrate. That's why fungi called hidden kingdom. They're hiding out. So once again, shelf mushroom attach itself to the usually rotten, dead, or dying trees. And in this particular case, the whole mycelium will go through that particular tree, and producing it will be kind of producing those fruiting body. Fruiting body, also known as a basidiocarp, right, or sporocarp. So here we're looking again at the cap or mushroom, sporocarp. This is the cap, that's its type. And you're looking at the gills, but again, that's if the question will say, name the structure, the correct answer would be a basidiocarp. So here we're looking at the gills. Again, basidiocarp, or from different previous basidiocarp again. So here we go. The whole thing is the basidiocarp. Inside of it, in the gills, it will have that club-looking structure producing spores, and we'll see it momentarily. So here we're looking at the cross section, of course, and then there will be a this is the spores produced. Here we go. So gills, right? And then the black dots are spores. And whenever we're looking under a higher magnification, you can see that bluish structure. So gill, bluish structure will be a basidium. That one is right there. And on top of the basidium, you will end up with the spores. Here we're looking at another basidium and the spores. The black dots are spores, of course. So yeah, you can see basidium and the spores. Now let's take a look. Ascomycota, similar. Actually, it should be. That's the Earth star. I think it's basidium. I need to double check. I will not put that one on the test. I think that's the actual basidio spore. Basidio mycota. This one is for sure. Marcella is actually Asca mycota, and whenever you're looking at the cross section, you will see again tons of spores in a very specific structures. The structures looks like a sack filled up with the spores. So those purple bluish dots, dark dots, that's the spores within the sac. That's why it's called sac fungi, ascus, Ascomycota. Ascus means sac. So here we go. So in this particular case, be able to what type of reproduction do those fungi do? That's a sexual reproduction. And Here we have Marcella, yeah, ascus. Ascus is the whole structure, and ascospore is one of those round purplish things. All of the spores are haploid once again. Yeah. Now we're looking at the Deuteromycota imperfect fungi. What type of reproduction do those fungi do? Asexual reproduction. Which one they do not do? As far as we know, we never saw them doing the sexual reproduction. And all of those particular bluish tealish strands are nothing else but the mycelium, right? Now we're looking at the mycelium, which start producing asexual sporangium, also known as a conidiophores, right? Conidiophores, and those tiny little bitty dots are conidiospores. So you can clearly see them under a higher magnification. So tons of spores, tiny little bitty. All of those strands, however, that's the mycelium, right? So you need to be able to recognize mycelium, the bluish tealish part, the round lollipop thingy known as the conidiophore, and the tiny little bitty dots on that. You can clearly see it here. That would be conidiospores. That's asexual reproduction, but spores continue to be haploid. Yeah, you can see that. Conidiophore with the conidiospores coming out again. I took the picture of the penicillin last time I was in the lab, but I will not be put it in this lab practical. Now, whenever we're looking at the lichens, you need to know three types: the crusty ones, cruticos, then that looks like a crust, right? The leafy ones, folios, more folios, and then the kind of bushy ones, that's known as the fruticos. Yes, you can see that the fruticos right here and the folios right there. Again, we have a bunch of folios and the fruticos together at on the same branch. So crustos, folios, and fruticos. So make sure you understand that any kind of lichen is made out of two organisms. That's a symbiosis. We have a mycelium of the fungi with embedded algae, and those algae gives the different color to the actual lichen. So what two organisms make up of the lichen? Fungi and the algae. What type of the symbiosis? The type of the symbiosis will be mutualistic or mutualism. Quid pro quo. I'll give you this, you'll give me that. Okay, so they mutually benefit. And describe benefits of two organisms. So algae provide food, glucose to the fungi. Fungus provide the protection as well as the inorganic compounds such as water and some of the minerals to the algae. Together, they live fairly happily, known as the pioneer species. They literally can live on a bare rock. In this case, they are on the branches. They're not parasitizing. They need just a place to live. That's between the lichen and the tree. It's something which is known as a commensalism, because tree doesn't really care, and the lichen by itself is benefiting by having the place to live. And we are pretty much done for today with the review. Let's see. Stop record.